Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 4 A-Level Structured Question May June 2021 Paper 4-3. Today we'll be solving from question number 1 to question number 5. Figure 1.1 is a diagram of a part of a sarcomere in a striated muscle. Alright, we can see the actin, we can see the myosin. So obviously D should be myosin head. We can also see the A, B, and C structure. So the question says, with reference to figure 1.1, name A, B, C, D. So we can see A is basically the site where the myosin head can bind, so it will be the binding site. B is tropomyosin, C is troponin, and D is myosin head. Part B. When a muscle cell is stimulated, calcium ions are released from the sarcoplasmic reticulum. With reference to figure 1.1, describe the role of calcium ions in the contraction of sarcomere. So we know calcium ions is released from the sarcoplasmic reticulum. Calcium ion then binds to the troponin and causes the tropomyosin to move and expose the binding site. The myosin head then binds to the actin, forming a cross bridge between the two types of filament. C. When a mammal dies, aerobic respiration stops. The striated muscles contract and remain contracted for a few hours after death. Suggest why the muscles remain contracted for a few hours. So when aerobic respiration stops, very little ATP is produced. So the myosin head cannot be released. Question number 2. During respiration, respiratory substrates such as glucose are used as a source of energy to produce ATP molecules. Cells can maintain a different concentration of ions within the cell compared with the extracellular environment. It is estimated that animals may use 50% of ATP made from respiration to maintain higher concentration of some ions inside cell. Name the mechanism that can maintain a higher concentration of ions inside a cell. Active transport. Active transport can maintain higher concentration of ions inside cell. 2a part 2. Only a proportion of energy released by the respiration of glucose is used to make ATP. The energy yield from respiration of glucose in aerobic respiration is 2870 kJ per mole. When ATP is hydrolyzed to ADP, it releases 30.5 kJ of energy. It is estimated that 31 moles of ATP are made per mole of glucose respired. Calculate the percentage efficiency of glucose respiration. Show your working. So basically 31 ATP is produced. Each ATP releases about 30.5 kJ per mole energy, which makes it 945.5 kJ. Now percentage efficiency is equals to 945.5 divided by the total amount of heat energy released, 2870, into 100%, which gives us a result of 33.94. Now since our answer has to be given in whole number, so it will be 34. So 34%. The energy that is not converted to ATP during respiration is released as heat energy. State one homeostatic use of heat energy in mammals. Well, in mammals, heat energy is used to maintain constant temperature. Part 4. State the term used to describe the reaction that results in production of ATP during the Krebs cycle. So during the Krebs cycle, production of ATP occurs in substrate link phosphorylation. So the answer will be substrate linked phosphorylation. Part B. When a person exercises, power is generated. Power is measured in joules per second. The power generated when a person exercises will vary depending on the type and intensity of exercise the person is doing. More power is generated when the intensity of exercise increases. An experiment was carried out to determine whether increasing the intensity of exercise in healthy human male causes a change in the value of respiratory quotient or RQ value. The man had a balanced diet. The RQ was calculated at rest. He carried out exercise for the same length of time on 4 separate days. The intensity of exercise was increased over the 4 days, generating different powers. The RQ was calculated for the 4 different powers generated. The results are shown in figure 2.1. So we can see power generated as it increases. RQ value also increases, goes beyond 1, which actually RQ value beyond greater than 1 indicates anaerobic respiration. Now the question says, with reference to figure 2.1, suggest what can be deduced from the RQ at rest. So basically RQ of 0.8, yes, RQ of 0.8, a mix of respiratory substrate, example lipid, carbohydrate and protein, are being respired. RQ when the power generated is 110 joule per second. So at 110 joule per second, as you can see, it's 1. So respiratory substrate is carbohydrate only. RQ when the power generated is 200 joules per second. When the power generated is 200 joules per second, anaerobic respiration is taking place, because we can see a value of RQ of 1.2, so which is indicative of anaerobic respiration due to low oxygen availability. Part C. Explain why a person continues to breathe deeply and at a higher rate for some time after the person has stopped exercising. Well, when a person continues to breathe deep and at higher rate after exercise stops due to oxygen depth, the extra oxygen is needed to convert lactate to pyruvate, to convert lactate to glycogen. The extra oxygen is also needed to reoxygenate hemoglobin. The extra oxygen is needed to support a higher metabolic rate, which is often the case after a very intense exercise. The metabolic rate stays very high. Question number 3. Fruit flies of the genus Bactrocera are pests that damage fruit crops. Table 3.1 gives the names and geographical ranges of Bactrocera fruit flies that were classified as 4 separate species. First of all, name B. dorsalis, China, India and Thailand. B. invadens, Africa and India. B. papaya, Indonesia, Malaysia. B. philippinensis, Philippine Island and Borneo. In 2014, the classification of these flies was changed. All 4 species were recognized as belonging to a single species, B. dorsalis. Suggest reason why 4 species were originally classified as separate species. Well, the 4 species have differences in morphology and also they may have differences in physiology. The 4 species have genetic differences and there is difference in their behavior or the ecological niche that they occupy. The 4 species are also found in different geographical areas, which gives the scientists idea that they were separate species. Next question. Some student decided to investigate whether the flies were members of one species or 4 separate species. Suggest a simple investigation that the student could carry out. The student can mate them together and see if the offspring that they produce are fertile. The student can collect DNA samples and match them using bioinformatics investigation as well. Part B. Female bee dorsalis lay their egg in different types of fruit, such as avocados, bananas, mangoes, and papayas. The eggs hatch into larvae that eat the fruit. The action of the female flies and larvae allows microorganisms to enter the fruit. The microorganisms feed by secreting extracellular enzyme causing the fruit to rot. Name two kingdoms that include organisms that could spoil fruit by secreting extracellular enzymes. So basically, fungi releases extracellular enzymes and they can cause fruit to rot. And also prokaryote, they can release extracellular enzyme. Prokaryote. And finally, protoctis can also be there. Protoctis. Before the four Bactrocera species were reclassified as a single species in 2014, some governments in Asia banned fruit imports from African countries to avoid introducing bee invaders as an alien species. Explain why the introduction of alien species should be avoided. Alien species may easily become invasive since alien species usually don't have any natural predators in the places where they're introduced. Alien species may compete with the local species for food and for place to live. Alien species may decrease biodiversity. Alien species may disrupt food web, and alien species may also introduce diseases as a result of their uh, daily activity. Guys, to receive a full mark in this particular question, you only require 4 marking points. If you notice carefully, this question consists of 1, 2, 3, 4, 5, and 6 marking points. Now, if you want to memorize all 6 of them, very good. If you don't want, then at least choose 4 which you find relevant towards this particular answer and then write it in your copy and memorize them. Part D. Suggest how reclassifying bee invaders as bee dorsalis will benefit fruit producing countries in Africa. So basically, reclassifying bee invaders as bee dorsalis will allow to lift the ban on African fruit imports. African countries will be able to export more fruit to Asia, and this will bring more economic benefit and work opportunities for the African farmers. Question number 4. A. BT maize has been genetically engineered to resist insect attack in 2019. BT maize formed 83% of the maize grown in the USA. Outline how the genetic engineering process produced BT maize plants with resistance to insect attack. The gene that produces BT is cut from Bacillus thuringiensis bacteria using restriction enzyme. The gene is then cloned using polymerase chain reaction. The gene is then inserted into plasmid and a promoter is also added to ensure gene expression. The plasmid is then sealed using DNA ligase. So DNA ligase basically seals the plasmid. Once the plasmid is sealed, the maize embryo takes up the plasmid containing recombinant DNA. The maize then produces Bt toxin by expressing the new gene. Agrobacterium tumefaciens can also be used to deliver the recombinant plasmid into embryo. Part B. A proposed conservation benefit of BT maize is that unlike chemical insecticide sprays, it should not harm non-target species. When BT maize was first grown in USA, scientists were concerned that the maize pollen containing the Bt toxin might be deposited by the wind on wild plants such as milkweed, Asclepias spp. Milkweed leaves are the food sources of caterpillar larvae of the monarch butterfly, Danus plexippus. In 1999, a laboratory experiment tested the effect of feeding milkweed leaves treated in different ways to the caterpillars of monarch butterflies. Each group of caterpillars was provided with milkweed leaves to eat throughout the experiment. The 3 different treatments were leaves with no pollen, leaves dusted with pollen from non-BT maize, leaves dusted with pollen from BT maize. And figure 4.1 shows the results of the laboratory experiment. We can see percentage survival of monarch caterpillars. Leaves with no pollen, there is 100% survival. Leaves with pollen from non-BT maize, there is also 100% survival. We can see that. However, leaves with pollen from BT maize, we can see if the pollen from BT maize are taken, then we can see that actually the monarch butterflies caterpillars actually decreases in number. State what can be concluded from figure 4.1. So basically we can say pollen from BT maize does kill the caterpillars. Pollen from non-BT maize does not harm caterpillars, and eating leaves with no pollen does not kill caterpillars as well. As the number of days of eating pollen from BT maize increases, more caterpillars are actually dying. Part 2. Laboratory results are not always useful in predicting what will happen in real ecosystems. Some scientists predicted that increasing the area of BT maize in the USA would decrease the number of monarch butterflies, based on the result in figure 4.1. More monarch butterflies were counted in 2019 than in 1999, even though the quantity of BT maize grown more than tripled. Suggest reasons why the predicted decrease in the number of monarch butterflies did not occur in the real ecosystem. Well, it can happen that BT pollen did not get deposited onto the milkweed plants. The pollens might be released at different time to when caterpillars actually feed on the milkweed plants. Caterpillars develop resistance to BT toxin. In 2019, breeding conditions were better than in 1999, which can easily be the case. Example, herbicides may have reduced milkweed availability. In maize field, caterpillars and BT maize might not be located in the same areas. So which is why monarch butterflies which may have increased in number. Question number 5. Photosynthesis is a process that results in the synthesis of complex organic molecules. Respiration is a process that involves the breakdown of complex organic molecules. The light-dependent stage of photosynthesis involves cyclic and non-cyclic photophosphorylation. Describe two differences between cyclic and non-cyclic photophosphorylation. So basically, cyclic photophosphorylation involves only PS1 and non-cyclic photophosphorylation involves PS1 and PS2. Cyclic only produces ATP and non-cyclic phosphorylation produces ATP and reduced NADP. Part B. Biochemical processes involving carbon dioxide change the external environment of the aquatic plant. Elodia canadensis and the pond snail Limnaea stagnalis. Carbon dioxide dissolves in water to form carbonic acid. A student set up 8 test tubes. Each test tube contains 30 cm3 of distilled water containing a pH indicator bromothiamyl blue. Figure 5.1 shows the experimental setup for these 8 test tubes. The test tubes were left for 12 hours. So we can see highlight intensity. Alright, there is an empty test tube. There is a test tube with this aquatic plant. There is a test tube with pond snail. There is a test tube with aquatic plant and pond snail. So the same process is repeated in the dark. Now let's carry on. Figure 5.2 shows how the color of bromothiamyl blue changes with pH. Alright, so first of all, let's indicate where photosynthesis will occur. So we will have photosynthesis here. In dark, there will be no photosynthesis. There will only be respiration here. And there will be respiration and photosynthesis here. There will only be respiration. There will only be respiration in 6, 7, 8 in all of them. Only respiration will occur here as well. Okay, let's carry on. Now, we can see blue indicates increase in pH, green indicates neutral pH, and yellow indicates decrease in pH. Remember, decrease in pH is going to occur in increase in CO2. Increase in pH is going to occur when there will be a decrease of CO2. And neutral pH will be when there will be no increase or decrease of CO2. So all tubes were green at the start of the experiment. The results of the experiment are shown in table 5.1. We can see 1 green, blue, yellow, green, green, yellow, green, yellow, yellow. Okay. Explain the purpose of test tube 1 and test tube 5. So test tube 1 can be a control and test tube 5 can be a control. So they can act as a control. Explain the results of test tube 2, 3, 4. Test tube 2. Plant carries out photosynthesis and uses up CO2 in the Kelvin cycle. Test tube 3. Alright. In test tube 3, obviously the test tube 3 there is, you can see yellow. So blue indicates high pH and yellow indicates low pH and green indicates neutral. So in test tube 2, alright, we can see only photosynthesis is going to take place. From the previous diagram, we can see only photosynthesis is going to take place. So carbon dioxide concentration is going to decrease. And in test tube 3, snail carries out respiration, which produces carbon dioxide in Krebs cycle, and CO2 reduces the pH. So we can see in test tube 3, only respiration occurs. In test tube 4, both respiration and photosynthesis is going to take place. Alright, and probably it's going to take place at the same rate because CO2 produced in respiration was used in photosynthesis. That's why test tube 4 remained green, which is neutral. Part 3. Explain the color change in test tube 6. So basically test tube 6 is yellow green. So less photosynthesis, so less carbon dioxide is used. In dark, no light dependent stage can occur, so no Kelvin cycle to fix CO2. Respiration does not occur producing CO2. Part 3. Explain the color change in test tube 6. So color change in test tube 6, we can see that here there is no photosynthesis, only respiration occurs. Alright, so respiration does occur producing CO2 and respiration rate is slower in aquatic plants, so slight decrease in pH will be observed in test tube 6. Guys, that's all for today's video. See you in the next video where we will be solving question number 6 to question number 10. And if you like our video, please like the video and subscribe to our channel and also share it with your friend if it helps you. Alright, see you in the next video. Bye bye.